Dear students, welcome to the session of Massive Open Online course. In this session, we will study Chlamydomonas. First, objectives. To understand systematic position of Chlamydomonas, to study its distinctive features, to know the methods of its reproduction, to study its life cycle, to realize the evolutionary significance of its sexual reproduction. First, a brief account of its structure. Chlamydomonas is a minute unicellular eukaryotic organism seen in aquatic habitats. It has a biflagellate ellipsoid cell surrounded by a well-defined cell wall. It generally possesses a cup-shaped chloroplast in its cytoplasm. It shows phototactic and chemotactic locomotory movement in water, which occurs with the help of two whiplash flagella of equal length and positioned at anterior end of the cell. Now it is systematic position. Chlamydomonas occupies a controversial systematic position. Being unicellular eukaryote, it should have been placed in kingdom Protista, as all unicellular eukaryotic organisms are nowadays placed in single kingdom Protista. However, due to its close resemblance with plants, most biologists place it in division Chlorophyta of kingdom Plantae. Therefore, it belongs to kingdom Plantae, division Chlorophyta, class Chlorophyceae, order Chlamydomonidales, family Chlamydomonidaceae, genus Chlamydomonas. Now its habit and habitat. Chlamydomonas is a large genus including 500 species adapted to different habitats. Most species are seen in freshwater habitats rich in nitrogen and organic matter. Some species occur in ponds, pools, water tanks, sewage tanks, and in slowly running water. Chlamydomonas grows on the surface of water and it makes green. Some species are terrestrial and seen in moist soil. Few species like Chlamydomonas halophila, Chlamydomonas ehrenbergii are seen in brackish water. Chlamydomonas nivalis grows on snow and imparts a red color to snow called red snow with the help of its hypnospores, which contain a red pigment called hematochrome. Chlamydomonas yellowstonensis, reported from snow in Yellowstone Park of America, imparts green color to snow. Now its shape and size. Chlamydomonas is a unicellular alga. It has ellipsoid, ovoid shape having round base and pointed end. It is 20 to 30 micrometers in length, 20 micrometers in width. The pointed end has two flagella. Now structure. Chlamydomonas cell consists of cell wall followed by plasma membrane, cytoplasm, eye spot and flagella. First, cell wall. It's a thin, firm, cellulosic covering of cell. In some species, it remains surrounded by a gelatinous pectic sheath. Plasma membrane. It's a thin, phospholipoproteinaceous covering of cytoplasm lying inside cell wall. Now cytoplasm. It is the colorless, colloidal, aqueous, living solution lying inside plasma membrane. It contains non-membranous organelles like centrosome and basal bodies called biliferoplasts. Single membrane bound organelles like endoplasmic reticulum, dictyosomes, contractile vacuoles, and double membrane bound organelles like mitochondria, a chloroplast and nucleus. Now, chloroplast. It is a single cup-shaped, possessing a proteinaceous body called paranoid at its center, which plays key role in polymerization of glucose to starch. Most species have single chloroplast, but few species like Chlamydomonas deberniana have two or more. Chloroplast contains chlorophyll A and B. Paranoid is absent. Cell has two contractile vacuoles which lie at the base of flagella and play a role in excretion and osmoregulation. Now, flagella. The anterior end of the thallus has two flagella. Both the flagella are of whiplash type, also called acroneumatic. They have equal size. The flagella are mostly longer than the thallus, that is cell, but in some species they can be equal or shorter. Each flagellum originates from a basal granule called biliferoplast and comes out through a fine canal in cell wall. Now, neuromotor apparatus. In some species of Chlamydomonas, like Chlamydomonas nasuta, a sensitive neuromotor apparatus is present. It controls movement of thallus in response to light, chemicals, and other stimuli. It consists of two basal granules called biliferoplasts, from which the flagella originate. It also has a transverse cytoplasmic fiber called paradesmus, which connects two biliferoplasts, and a cytoplasmic fiber called rhizoplast, which connects one biliferoplast with the centrosome, and a small delicate fiber connecting centrosome with nucleolus. Now, shape of chloroplast. Chlamydomonas generally has a large cup-shaped chloroplast, parietal in position in its cytoplasm towards the basal end. But it can be of various shapes in different species. The chloroplast is H-shaped in Chlamydomonas basileata. It is reticulate in Chlamydomonas reticulata. It is parietal in Chlamydomonas nucicola, stellate in Chlamydomonas arhene, and axile in Chlamydomonas stinae. It is generally associated with a paranoid covered with starch plates, but sometimes paranoids can be more than one. The paranoids are two in Chlamydomonas deberniana and many in Chlamydomonas gigante. They play a role in polymerization of glucose produced through photosynthesis. In chloroplast, there are 2 to 6 thylakoids which join to form a granum. Now, stigma or eye spot. The anterior side of the chloroplast possesses a tiny region with orange or reddish color called stigma or eye spot. It serves as photoreceptor and plays a role in directing the movement of flagella that enable the cell to move from bright light to diffused light. The eye spot is made up of a curved pigmented plate. The plate contains 2 to 3 parallel rows of droplets or granules containing carotenoids. Now, reproduction in Chlamydomonas. Chlamydomonas reproduces asexually as well as sexually. Asexual reproduction, it occurs during favorable conditions and involves formation of zoospores, applanospores, hypnospores and palmella. First, zoospore formation. Chlamydomonas forms zoospores during favorable conditions. The protoplast contracts and gets separated from cell wall. The parent cell loses flagella. In some species of Chlamydomonas, flagella are absorbed. The contractile vacuoles and the neuromotor apparatus disappear. The protoplasm divides longitudinally by simple mitotic division, forming two daughter protoplasts. The second division of protoplasm takes place at right angles to the first, thus making four daughter cells. Sometimes the protoplasm may further divide to make 8, 16 and 32 daughter protoplasts. The paranoids and initials of neuromotor apparatus also divide. The contractile vacuoles also develop in daughter protoplasts. Each daughter cell develops a cell wall, two flagella and transforms into a zoospore. The zoospores are liberated from the parent cell or zoosporangium by gelatinization or rupture of cell wall. The zoospores are identical to parent cell in structure but smaller in size. They simply enlarge to become mature. Under favorable conditions, the formation of zoospores can take place every 24 hours. Now, applanospores. The applanospores are formed slightly under unfavorable conditions. For example, in Chlamydomonas caudata, the parent cell loses flagella. The protoplast rounds off and secretes a thin wall outside, but does not develop flagella. These non-motile structures are called applanospores. On the approach of favorable conditions, applanospores may germinate either directly or divide to produce zoospores, which give rise to mature Chlamydomonas cells. Now, hypnospores. It occurs during extremely unfavorable conditions. The protoplast shrinks, loses flagella, secretes a thick wall on its surface and remains inactive. The thick-walled, inactive and resistant spores are called hypnospores. It is seen in Chlamydomonas nivalis. The hypnospores are small like applanospores. On the approach of favorable conditions, they germinate and give rise to mature Chlamydomonas cells. Now, palmella stage. The palmella stage is formed under unfavorable conditions like shortage of water and excess of salts. The protoplast of parent cell divides to make many daughter protoplasts, but they do not form zoospores. The parent cell wall gelatinizes to make mucilaginous sheath around the daughter protoplasts. The daughter protoplasts develop gelatinous wall around them, but do not develop flagella. They are called palmella spores. They divide and redivide to produce an amorphous colony with indefinite number of spores. This mass of palmella spores enclosed within a gelatinous sheath is called palmella. When favorable conditions return, the gelatinous wall is dissolved. Palmella spores develop flagella and the spores are released to make new Chlamydomonas cells. This was about asexual reproduction. Now, sexual reproduction in Chlamydomonas. Sexual reproduction involves isogamy, anisogamy, or oogamy. There can be homothallism, that is, both the types of gametes are produced by the same cell, as seen in Chlamydomonas mogamme and Chlamydomonas media. Or there can be heterothallism, that is, positive and negative gametes come from different parents. The gametes may be naked, called gymnogametes, as in Chlamydomonas debariana, or covered by cell wall, called calyptogametes, as in Chlamydomonas media. First, isogamy. Most of the Chlamydomonas species are isogamous. In isogamous reproduction, the fusion of gametes which are similar in size, shape and structure take place. These gametes are morphologically similar but physiologically dissimilar. In many isogamous species, the vegetative cells may directly function as gametes without undergoing any division. For example, in Chlamydomonas snowi, this fusion is called hologamy. The thalli shed their cell walls and function as gametes. The two gametes come close to each other by their anterior ends, and later fusion proceeds to lateral sides. The fusion product is quadriflagellate initially and binucleated structure with two paranoids and two eye spots. The quadriflagellate zygote remains motile for several hours to few days. In some species, the vegetative cells do not shed their cell walls after the union of their contents. The contents of one gamete enter into another gamete as such. According to Chapman, the isogamous reproduction takes place by the production of 8, 16 or 32 biflagellated gametes. The process takes place as follows. The vegetative cell functioning as a gametangium comes to rest, loses its flagella. The protoplast withdraws itself from the cell wall. The protoplast divides by repeated longitudinal mitotic divisions to produce 8, 16, 32 or 64 daughter protoplasts. Each daughter protoplast develops a pair of flagella and transforms into a gamete. The gametes are liberated by breaking the cell wall of gametangium. The flagella of gametes are covered by agglutins and secrete a hormone called gamone. These chemical substances are involved in the recognition of gametes of opposite nature. In heterothallic species, positive and negative gametes cluster together, and this phenomenon is called clumping. The gametes of opposite strain fuse by anterior end, that is, apical fusion or lateral, that is lateral fusion. The paired gametes move away from the clump. The wall at the place of contact dissolves and fertilization takes place in two steps, namely plasmogamy and karyogamy. In plasmogamy, the fusion of cytoplasm, and in karyogamy, there is fusion of nuclei. After fertilization, a quadriflagellate zygote is produced. The zygote later on loses flagella and gets covered by a thick cell wall and is now called zygospore. Now, anisogamy. In anisogamous reproduction, the gametes are unequal in size but have same shape. The female gametes or macrogametes are larger. For example, in Chlamydomonas bruni and Chlamydomonas subugama. The macrogametes are formed in female gametangium, in which the protoplast divides to make 2 to 4 gametes only. The macrogametes are formed in male gametangium, whereas the protoplast divides to make 8 to 16 gametes. The microgametes are more active than macrogametes. The microgametes come close to macrogamete. The protoplast of microgamete enters into macrogamete, and after fusion, a diploid zygote is formed. The zygote secretes a thick wall and transforms into zygospore. Now, oogamy. The oogamous sexual reproduction takes place in Chlamydomonas cocifera and Chlamydomonas ooganum. The vegetative cell, functioning as a female cell, withdraws its flagella and directly functions as non-motile macrogamete or egg cell. The female gamete contains many paranoids. The microgametes are formed by 4 divisions of protoplast, as in case of anisogamous reproduction. The microgamete reaches the female gamete and unites by anterior ends. The contact wall between the two dissolves. After plasmogamy and karyogamy, a diploid zygote is formed. The zygote secretes a thick wall and transforms into zygospore. Now, let us see the structure of zygospore. The zygote is diploid, formed by the fusion of gametes. It secretes a thick wall, which is smooth or ornamented, and accumulates a large amount of oils and starch. This thick-walled inactive zygote is called zygospore. Zygospores are red in color due to presence of a pigment called hematochrome. It survives long period of unfavorable conditions and becomes active on the approach of favorable season. Its nucleus divides by meiosis to make 4 haploid nuclei. The 4 daughter protoplasts, each with 1 haploid nucleus, form 4 haploid zoospores called meiospores. Each zoospore contains neuromotor apparatus called eye spot, 2 flagella and 2 contractile vacuoles. In 4 zoospores, 2 may be positive and 2 negative type in heterothallic forms. The number of meiospores per zygospore are 8 in Chlamydomonas reinhardtii or 16 to 32 in Chlamydomonas intermediata. Now, life cycle. Chlamydomonas shows haplontic life cycle characterized by zygotic meiosis and formation of mitogametes. Hope you have enjoyed this lecture and you have learned from it. Thank you.